Hello everyone, now we are going to discuss about the important long answers in the zoology second year. In the zoology second year, the important long answers as chapter wise we are seeing now. From the first unit, if you are seeing diversity in the living world, from the first unit there is no chance of asking the important long answers particularly, but if they are asking from the first unit, there may be chance of asking about the biodiversity conservation. When they are asking about the biodiversity conservation for the 8 marks, we have to explain about the in situ conservation, ex situ conservation, both things here. In the in situ conservation, we have to explain about the national parks, sanctuaries, biosphere reserves and sacred groves, and for them all the examples we have to give. For the national parks, Jim Corbett National Park example we can give, Kasu Brahmandra Reddy National Park we can give. Then for the sanctuaries, we can give the Bharatpur Bird Sanctuary example we can give, and for the biosphere reserves, different examples we can give for the biosphere reserves. So totally 17 biosphere reserves. In that thing, the 17th biosphere reserve is the Seshachalam Biosphere Reserve. The sacred groves, particularly different examples we can give actually. Sarguja and Bastar, then Chanda, all these examples we can give for the sacred groves. And for the ex situ conservation, particularly, we have to explain about the zoo parks, in vitro fertilization, gene banks, and preservation in the nitrocellulose method. And these methods we have to explain for the 8 marks here, if in the first unit they are asking. And in the second unit, there is a lot of scope of asking the 8 marks here. In the second unit, the frequently asked, the mostly asked 8 marks question is the epithelial tissues. Whenever they are asking about the epithelial tissues, first we have to give the introduction of the epithelial tissues. What are the epithelial tissues? They are the surface lining tissues, and epithelial tissues, different types of epithelial tissues. If you are taking simple epithelium, we have simple epithelium consisting of a single layer of cells and only one type of cells. Simple epithelium, again we are dividing into simple squamous epithelium, which it is consisting of a single layer of cells. Then simple squamous epithelium examples we can take as actually the linings of the Bowman's capsule, we can take the example. And simple cuboidal epithelium, the name itself indicate actually in the simple cuboidal epithelium the cuboidal shaped cells are present. And the examples of the cuboidal epithelium includes the germinal epithelium, which is lining the proximal collimated tubule and distal collimated tubule. This is about the simple epithelium. And then columnar epithelium we are saying. If I take the columnar epithelium again we are dividing into two types mainly. Two types mainly consisting of what? Ciliated columnar epithelium, non-ciliated columnar epithelium. Ciliated columnar epithelium clearly indicates actually the columnar type of cells which are consisting of cilia on them. Example, particularly we can take for this thing actually ventricles of the brain we can take, then spinal cord we can take the example. Then for the non-ciliated columnar epithelium, particularly if you are taking in the non-ciliated columnar epithelium, name itself indicates cilia is absent, only columnar shaped cells are present here. The example we can take for this thing, particularly intestines and microvilli, we can take the examples for particular these things here. Then compound epithelium, name itself indicate here compound epithelium, it means it is consisting of a different types of cells. Different types of cells and different group of cells, they are arranged in a tissue. That thing we are calling as what? Compound epithelium we are calling. In the compound epithelium again, the dry skin is always lined by the stratified keratinized compound epithelium, and the moist skin like esophagus, pharynx, they are lined by the stratified non-keratinized epithelium. Like this we can explain for the compound epithelium. Then glandular epithelium we are saying, glandular epithelium clearly says that actually it is consisting of a glands which secretes the secretions. Glandular epithelium again we are dividing into unicellular and then multicellular, which unicellular consisting of a single type of cells, and multicellular consisting of many type of cells. Apart from this thing, exocrine glands, endocrine glands we had. Exocrine glands, as we know, they are the ducted glands. Examples we can take a salivary glands and all these things we can take the example. Endocrine glands are the glands which are ductless glands, and mainly important point actually endocrine glands always produces the hormones. Pituitary gland, thyroid gland, all these things we can take the example of the endocrine gland. And mainly the exocrine glands which we had, particularly exocrine glands again we are dividing into merocrine, apocrine, holocrine. Merocrine example we can take particularly as a pancreas. Apocrine example we can take as a sweat glands. Then lastly holocrine example we can take as a sebaceous glands. All these things we have to explain in the epithelial tissues, and diagrams we have to draw compulsory wherever we are required. This is about the epithelial tissues. Sometimes if they are not asking the epithelial tissues, they may ask about the coelom and symmetry. Simple answers, coelom and symmetry. Coelom is a body cavity which is present coelom is a fluid which is present between the body cavity and then body wall particularly. And coelom, different types of the coeloms we had: acoelomates, pseudocoelomates and schizocoelomates. Three things we have to explain. Schizocoelomates, again we have to explain about the formation of the schizocoelom and enterocoelom, and the eucoelomates we have to explain, and diagrams we have to give the examples here. Then symmetry. Symmetry is the phenomenon of arrangement of the organs in the body in equidistance. That is the symmetry we are saying. Symmetry, again we are dividing into asymmetry, then radial symmetry, bilateral symmetry. Asymmetry example we can take as a sponges, which they are not showing the symmetry at all. And radial symmetry is a symmetry in which what is happening, we can divide the organism into two equal halves by only one plane passing by any plane passing through the central axis. That is the radial symmetry. And bilateral symmetry is a symmetry in which what is happening, we can divide the organism into two equal halves passing through the central axis by only one plane, we are saying. Example of the radial symmetry, we can take it as example as a hydra. For the pentaradial symmetry, we can take it as example as a starfish or asterias. Asymmetry is sponges, we took the example, and bilateral symmetry, we can take the example as a higher organisms, we can take the example. These are the three questions particularly in the second unit. When we are going for the third unit, actually invertebrate phyla or animal diversity 1, we are saying in this particular things. Actually earlier they are not asking from the invertebrate phyla, but particularly as of 70% and 30% syllabus, they are asking about the Ferretima digestive system they are asking, and Ferretima reproductive system they are asking. If the Ferretima digestive system they are asking particularly, we have to draw a diagram particularly, and then we have to indicate the parts and in which segments particularly which parts are present, we have to clearly indicate here. 1, 2, 3 segments buccal cavity, 4th segment pharynx, 5, 6, 7 esophagus, 8 segment gizzard is also called as a grinding mill, 9 to 14 segments stomach, 14 to last intestine, and 26th segment always consisting of intestinal caeca, and 26th to last we have tiplozole. Tiplozolar region is a region which always increases the area of absorption in the intestine. About all these particular segments and parts we have to explain in the digestive system of the earthworm, and process of digestion also we have to explain in the digestive system of the earthworm. If you are moving for the reproductive system of the earthworm, as we know, the earthworm is always, or Ferretima is always, Ferretima is the scientific name of the earthworm particularly. Ferretima is always bisexual. That means male and female reproductive organisms are present in the same organism, particularly the same Ferretima. In these things, same as in the digestive system segments which we have taken, same particularly which parts are present in which segments we have to clearly indicate particularly. First of all, 6th, 7th, 8th, 9th segments, 4 segments consisting of a pair of spermathecae. Spermathecae always stores the sperm. It is the structure of spermathecae consisting of ampulla and diverticulum, and 4 pairs of spermathecae are present particularly. Then a pair of testes are present particularly. 10th and 11th segments particularly, they are consisting of 2 pairs of testes particularly, and 11th and 12th segments are consisting of a seminal vesicles. Then from the 11th 12th segment to 18th segment, actually vasa efferentia is running particularly, and 18th segment is consisting of male genital pore. If you are taking the female reproductive system particularly, a pair of ovaries are present in between the 12th and 13th segments, and 14th segment is consisting of a oviduct, and same the 14th segment is consisting of a female genital pore. About all these parts we have to write, and we have to explain about the reproductive system and how the particularly this earthworm is participating in the copulation, how the earthworm is producing the larva, and all these things, molting, how it is taking place. Molting, it means changing, that we have to write in the reproductive system of the earthworm. These 2 questions are from the third unit, that is actually an animal diversity 1 or invertebrate phyla. From the 4th unit, there is no chance of asking the long answers, but if they are asking, they can ask the characters of the amphibians, reptilians, apes and mammals, they can ask particularly. We have to concentrate on the characters here. And 5th unit is not there in our syllabus, that is actually locomotion and reproduction. 6th unit, biology and human welfare, very very important unit. That means the life cycles which are there, they are present in this unit. From the 4 life cycles, 1 life cycle will be compulsory in the examination. And if you are seeing the 4 life cycles, first one is Entamoeba histolytica. Entamoeba histolytica life cycle, if you are taking, Entamoeba histolytica is a monogenic parasite which is completing its life cycle in only one host, that is human being. If you are seeing the life cycle of Entamoeba histolytica, different stages we are getting particularly: tropozoite stage, pre-cystic stage, cystic stage. These 3 are the main stages we are saying. The tropozoite stage is a stage, particularly it is a feeding stage. In the tropozoite stage, if you are seeing the structure of the tropozoite particularly, it is consisting of ectoplasm, endoplasm, then cytoplasm, food vacuoles with RBC, cartwheel shaped nucleus, ribosomes, all these are present in the tropozoite. This tropozoite, it is giving rise to next stage, that is actually pre-cystic stage. In the pre-cystic stage, cartwheel shaped nucleus is present and glycogen granules are present, 2 are present, apart from all the parts here. Pre-cystic stage, by developing the cyst wall, what will happen? It is converting into cystic stage, right? Cystic stage is because of to avoid the unfavorable conditions which is there in the intestine particularly. And after the cystic stage, in the cystic stage, the nucleus which is present, cartwheel shaped nucleus, it undergoes the division continuously and forms tetranucleate cyst stage. This tetranucleate cyst stage which we had particularly, it is the infective stage in the Entamoeba sclerotica, that is infective stage of the amoebiasis. Tetranucleate cyst stage which we had particularly, it is entering into the moist soil, and whenever we are taking the contaminated food and water, when it is consisting of this tetranucleate cyst stage, that means whenever a person who is suffering from the amoebiasis or amoebic dysentery, when he is passing the fecus, his fecus is consisting of a tetranucleate cyst stage. This tetranucleate cyst stage, when it is going and it is mixing with the moist soil, all these things here, whenever it is entering into the food and water, whenever the food and water is contaminated with the tetranucleate cyst stage, it is entering into again the fresh human host. In the human host, tetranucleate cyst stage which we had particularly in the human body, again it is losing the cyst wall. After the losing cyst wall, we are calling as a excision. After the excision, what will happen? The four nucleus which we had, the four tetranucleate cyst which we had particularly, they divides and forms the 8 nucleus particularly. The 8 nucleus we are calling as a metacyst. And out of the 8 metacyst particularly, 1 metacyst is developing into tropocyte. Again the life cycle is continuing, right? And if it is not affecting on the intestine, we are calling as a extra intestinal amoebiasis, right? And in this particularly long answer of the life cycles, first we have to explain about the life cycle, then we have to write about the diagnostic methods and pathogenicity, all these things we have to write, and then prophylaxis also we have to write particularly in this thing. This is about the Entamoeba sclerotica. And Entamoeba sclerotica name itself indicate, actually Entamoeba is the name of the organism. Histolytica, it means the histolysine is the enzyme which always disintegrate the wall of the large intestine. Then only the blood cells which are entering into the food vehicle, then we will get the food vehicles with the arteries. This is about the Entamoeba sclerotica. Then we are moving with the Plasmodium. Plasmodium is a very important, compulsory, frequently asked question in the final examination. And in the Plasmodium, first we have to give the introduction of the Plasmodium. What is Plasmodium? Plasmodium is a parasite. And then Plasmodium, and when we are writing about the introduction of the Plasmodium, we are writing about the 4 types of the Plasmodium: Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malaria. About 4 things we have to write. And then about the Sir Patrick Manson, Charles Laverson, and then Sir Ronald Ross. And Sir Patrick Manson, about the he identified the Plasmodium in the blood of the mosquito, and Charles Laverson first said that actually mosquito is responsible for the transfer of the malaria. And then Sir Ronald Ross has been discovered the oocyst in the stomach wall of the mosquito, hence he got the Nobel Prize. He done his complete research in the Secunderabad, hence honoring his research, particularly Fever Hospital in the Corinthi has been named as Sir Ronald Ross Institute for the Research of the Tropical Diseases has been given. Then if you are going for the life cycles particularly, sporocyte is the infective stage of the malaria. Plasmodium life cycle, if you are seeing, Plasmodium is always diagenic parasite because it is completing its life cycle in the 2 hosts, that is the primary host and secondary host we are saying. Primary host is the host particularly in which what is happening here, sexual life cycle occurs. The primary host for the mosquito is the female Anopheles mosquito, particularly Plasmodium is the female Anopheles mosquito, and secondary host is a human being. First of all, we are seeing about the Plasmodium life cycle in the human being. Plasmodium life cycle in the human being, commonly we are calling it as schizogony because many schizon stages are present particularly. Plasmodium life cycle in the human being, again we are dividing into what? Dividing into different stages here. First time we are calling it as a hepatic schizogony. Then next we are calling it as a gametocyte formation. We are saying hepatic schizogony, which we had, again we are dividing it into exoerythrocytic cycle, pre-erythrocytic cycle, then erythrocytic cycle. 3 things we are saying particularly. In the Plasmodium life cycle in man, the life cycle of the Plasmodium always starts with the sporocyte. Sporocyte is the infective stage. Whenever the female Anopheles mosquito, which is consisting of a sporocyte stage, when it bites a healthy person, then what is happening? It is entering into the human body, that means human blood circulation particularly, and firstly it is not entering into the human blood circulation. That's why we are calling it as a pre-erythrocytic cycle. What is happening? The sporocyte which we had, sporocyte is first converted into tropocyte. Tropocyte again converts into schizont. Schizont divides and forms the cryptocytes. Formation of the cryptocytes is the last stage of the pre-erythrocytic cycle. After the pre-erythrocytic cycle, again what will happen? We are getting the next exoerythrocytic cycle in the liver. It will take place particularly. In the exoerythrocytic cycle particularly, cryptozoites are entering. Again the cryptozoites are turning into what? Tropozoite into schizont. Schizont again divides and forms the micrometacryptozoites, macrometacryptozoites. Remember here, micrometacryptozoites are attacking the RBC cells and they are continuing the erythrocytic cycle. Macrometacryptozoites are attacking on the fresh liver cells and they are continuing the liver cycle again. Here, particularly pre-patent period we are calling. Pre-patent period is for the 8 days particularly. Pre-patent period, it means the sporozoite which is entering first time into the blood. Again the appearance of the sporozoite in the form of the cryptozoites. The middle time we are calling as a pre-patent period. Then erythrocytic cycle we are saying particularly. Erythrocytic cycle is also known as Camellia-Golgi cycle because Golgi has been described about this cycle particularly. In the erythrocytic cycle, first metacryptozoite, it is attacking the RBC cell. Then RBC cell is turning into tropozoite into schizont, again it is turning into vacuole which we had particularly. In the vacuole, whatever the secretions which we had, secretions are moved to the end particularly, that means nucleus, all these things, and they form the signet ring stage. That means as a ring like appearance which we are getting, there is a signet ring stage we are getting. Signet ring stage again gives rise to what? Gives rise to amoeboid stage by acquiring the pseudopodia, and it is a temporary, because always remember Plasmodium is a non-motile. It doesn't consisting of any locomotory organelles. Temporarily it will take the pseudopodium. It is amoeboid stage. Again the pseudopodium is lost. Amoeboid stage is gone off. Again schizont stage we are getting. After the schizont stage, again what is happening here? Hemozoin granules we are getting. Schaffner dots, and then hemozoin. Schaffner dots actually breakdown of the particularly hemoglobin. We are getting the hemozoin granules. Hemozoin granules gives rise to a Schaffner dots, red colored dots which are forming in the blood cells. We are calling as Schaffner dots. The Schaffner dots again gives rise to merozytes. Merozytes gives rise to what? Micrometacryptozoites and macrometacryptozoites. By the Schaffner dots only symptoms will be appearing in the life cycle of the Plasmodium. And incubation period we are saying particularly incubation period will be for the 14 days. Incubation period, it means the time between entry of the sporozoite and appearance of the symptom. That is the incubation period. Lastly, by the Plasmodium life cycle in the man, lastly what we are getting? Male gametocyte and female gametocytes we are getting. And next we are going to discuss about the Plasmodium life cycle in the female Anopheles mosquito. It is a sexual life cycle. Now, what is the last stage we got in the man, particularly man of the Plasmodium, particularly male gametocyte, female gametocyte. Whenever the female Anopheles mosquito is biting a person which he had the male and female gametocytes, that male and female gametophytic stage is entering into the mosquito. In the mosquito, what is happening? We are dividing the sexual life cycle in the mosquito particularly into what? Gametogony, fertilization, formation of ukinite and oocyst, then sporogony. 4 stages we had particularly. Here particularly the mosquito life cycle we are calling as a Ross cycle because Ronald Ross has been discovered the oocyst here. Here particularly first gametogony we are saying. Gametogony, it means formation of the male gamete and female gamete from the male gametocyte and female gametocyte we are calling as a gametogony. Male gametocyte particularly which we had, it is giving rise to what? Male gamete. Female gametocyte which we had particularly is given to female gamete. Female gamete consisting of a 8 flagellated structure. Male gamete is directly giving rise to male gamete particularly. Here what is happening? As male gamete, female gamete which we are particularly, they are not in a similar shape. That's why we are calling as anisogamous fertilization. After the anisogamous fertilization, what is happening here? Formation of the ukinite takes place. Fertilization, it means what? The fusion of the male and female gamete we are calling as a fertilization. After the fertilization, what is happening? Vermiform ukinite is formed. Ukinite, it means the earthworm shaped ukinite will be forming here, shape of the ukinite particularly. Ukinite, by acquiring the cyst wall, we are calling as oocyst. And whatever the nucleus we have, nucleus divides continuously. Oocyst, which we had particularly, oocyst in the next step, what is happening? It is giving rise to sporoblast stage. Sporoblast stage, it means what? It means the nucleus which we had in the oocyst stage continuously they are dividing and form the sporoblast here. After the sporoblast stage, what is happening? The sporoblast which we are developing continuously in the sporoblast stage particularly, the round structure they are developing into sickle-shaped structure. The sickle-shaped structures we are calling as the sporozoites. After the formation of the sporozoite, the sporoblast wall will be ruptured and the sporozoites will be released into the salivary glands of the mosquito here. Right? And this is the Plasmodium life cycle in the mosquito. Lastly, sporozoites are released. Sporozoites are the infective stage, and again whenever the sporozoites are entering into the new host, then again the life cycle is continuing. This is about the Plasmodium life cycle. And the Plasmodium life cycle, again symptoms we have tried, chill fever, coma, all these things, symptoms we have tried. And Then diagnostic method, blood test we are saying, and prophylaxis, how we are avoiding the mosquitoes, all these things we have tried here. This is about the Plasmodium. Then if you are seeing about the next life cycle, there is Ascaris particularly. Ascaris is a monogenic parasite which is completing its life cycle in one host, that is a human being. Ascaris, it is actually included in the phylum Nematoda and class Phasmida. Here what is happening in the Ascaris, male Ascaris is actually curved tail and female Ascaris consisting of a straight tail. In the Ascaris life cycle, if you are seeing particularly male Ascaris, female Ascaris, whenever they are copulating, then what will happen, the first stage which we are getting, there is a mammulated egg stage we are getting. Mammulated egg which is consisting of a rippled surface, protein coat, lipid coat and rippled surface we are having. In the mammulated egg, actually Ascaris lumbricoides which we had particularly after the copulation, Ascaris lumbricoides, they are producing 2 lakhs of the mammulated eggs per day. Each mammulated egg which they are produced here, they are present in the moist soil. In the mammulated egg, rhabdiform larva is present. And the mammulated egg which is consisting of a rhabdiform larva, we are saying as a first stage of rhabdiform larva. The first stage of rhabdiform larva undergoes the molting and forms the second stage of the rhabdiform larva. And the second stage of rhabdiform larva which we had particularly, it is entering into the human being. In the human being, what is happening, with the help of the hepatic portal vein, first it is entering into the liver. From the liver, with the help of the postcavalian vein, it is entering into the heart. From the heart, with the help of the pulmonary vein, it is entering into the alveoli of the lungs. In the lungs, what is happening here? Second stage of rhabdiform larva which we are, it is undergoing the second molting, that means second change, and form the third stage of rhabdiform larva. This third stage of rhabdiform larva which we had particularly, this third stage of rhabdiform larva is entering into the again human being, labium, pharynx, larynx, esophagus, then lastly it is entering into the visiting intestine. In the small intestine, what is happening? Fourth molting takes place and fifth stage of the larva is formed, and fifth stage of the larva will be developing into adults. This is the life cycle of the Ascaris. And in the Ascaris lumbricoides, we have to write about all these steps, and prophylaxis, diagnostic methods is common. And Ascaris always causes ascariasis disease. For the ascariasis, what is happening here particularly? It means severe stomach pain and nutritional deficiency are the symptoms. And prophylaxis, we are saying how we can avoid the Ascaris lumbricoides, that means parasites. It means same prophylaxis for the entamoeba and Ascaris lumbricoides, that we have to take the fresh fruits and fresh vegetables. We have to maintain the hygienity in our surface, in our surroundings particularly, and always we have to take the fresh foods only. We have to maintain hygienity, neatness we have to maintain. All these things we have to do to avoid the Ascaris lumbricoides parasite. This is about the Ascaris. And Eukaryaria bank of tea, if you are taking particularly, same Eukaryaria bank of tea also belongs to Nematoda and class Phasmida we are saying. Eukaryaria bank of tea also whitish color it is actually. Eukaryaria bank of tea, which we had particularly life cycle, if you are seeing, Eukaryaria bank of tea, diagenic parasite. It is completing its life cycle in the two hosts, that is actually in the female Culex mosquito and then man. Here particularly man, in the man, sexual life cycle of the Eukaryaria bank of tea takes place. In the female Culex mosquito, asexual life cycle takes place here. Whenever we are discussing about the Eukaryaria bank of tea particularly, Eukaryaria bank of tea also same thing. Male and female Eukaryaria, they are undergoing the copulation and they forms the particularly sheathed microfilaria larva. The sheathed microfilaria larva which we had particularly, the sheathed microfilaria larva which is present in the lymph glands of the particular human being, the sheathed microfilaria larva will be there in the human being for the 70 days. If it is not transferred to the next host for the 70 days, it will be dying. The sheathed microfilaria larva always exhibits a very, very important phenomenon, that is the nocturnal periodicity. Nocturnal periodicity, it means what? It means the sheathed microfilaria larva which we had, the time between 10 p.m. night 10 p.m. to 4 a.m. it is entering into the peripheral blood circulation to make adjustment for the female Culex mosquito to take this sheathed microfilaria larva, right? That is the nocturnal periodicity we are saying. Sheathed microfilaria larva is converting into what? Converting into particularly sausage shaped larva. Sausage shaped larva is converting into first stage of the larva. This first stage of the larva which we had particularly, it is undergoing the first molting and forms the second stage of the larva. Second stage of the larva is undergoing the second molting and form the third stage of larva. This third stage of larva is the infective stage in the Eukaryaria bank of tea. Third stage of larva is entering into the labium of the particular mosquito. In the labium of the mosquito, in the mosquito stomach wall, again what is happening here? Third stage of larva undergoes the third molting. Fourth stage of larva undergoes the fourth molting. Fifth stage of larva is developing into adults, and then again the adults are copulating, life cycle is continuing. This is about the Eukaryaria bank of tea. In the Eukaryaria bank of tea particularly, we have tried the pathogenicity of the Eukaryaria bank of tea is very, very important particularly. The pathogenicity, we have tried about the lymphadenitis, all these things we have to write. That means how the swelling of the lymph glands, all these things takes place, and all these things we have to write. Lymphoedema, all these things we have to write in the pathogenicity. And for the prophylaxis, particularly Eukaryotic bankruptcy is causing the elephantiasis, that means swelling of the legs, and all these things we are calling as elephantiasis, or filaria we are saying particularly. Prophylaxis, that means avoidance of that thing, Eukaryotic bankruptcy. If you avoid the mosquito, we can avoid the Eukaryotic bankruptcy. How we can avoid the mosquito? First of all, we have to avoid the stagnant water in our surroundings. Apart from that thing, what is happening? Whenever the stagnant water is present on the surface, we have to spray the kerosene or DDT, dichloro-diphenyl trichloroethane, all these things, and we have to use the mosquito mats, and we have to use the mosquito repellent creams to avoid the bite of the mosquito. This is about the Eukaryotic bankruptcy. Then if you are moving for the last unit, that is the ecology. In the ecology, most of the syllabus is deleted, but temperature is an ecological factor is a very important answer. Particularly the temperature is ecological factors, we have to discuss, we have to write particularly about how temperature is helping as a ecological factor in that thing. Stratifications we have to explain first. Summer stratification, winter stratification. Stratification, it means what? Changing in the lake ecosystem, changing of the layers. Epithelium, thermocline, hypolimnion, 3 layers we had. Epithelium consisting of temperature up to 21 to 29, particularly 29 degrees Celsius. And after the epithelium, then we are going down level, 9 degrees Celsius, that is a thermocline. Last level we are calling as a hypolimnion, 7 degrees Celsius, particularly 4 degrees Celsius we are saying. In the summer stratification, we are getting these 3 layers stratification. That means in a lake ecosystem only, if you are getting the 3 layers different temperature, we are calling as a stratification, summer stratification. In the winter stratification, what is happening? Same 3 layers are formed, epithelium, thermocline, hypolimnion. But what is happening here? Upper surface is always consisting of 4 degrees Celsius. Lower surface, when we are going down in the winter season, what is happening? Temperature is increasing. Whenever the overturn takes place, after the winter, whenever the next overturn takes place, what is happening actually? Slowly the downer layer water will be coming upper. That phenomena we are calling as a winter overturn. Summer stratification, winter stratification, we have to explain about the Van-'t-Hoff's rule. By increase of the every 10 degrees centigrade, what will happen actually? 10 degrees Celsius temperature, metabolic activities will be doubled. That we have to explain about the Van-'t-Hoff's rule. Allen's rule, and Benjamin's rule also we have to explain the temperature of ecological factor, and conformers, regulators, all these things we have to explain. Allen's rule and Benjamin's rule always explains about the body volume ratio, and then to avoid the heat loss, short earlobes and short limbs should be there. That will be explained by the Allen's rule and Benjamin's rule, all these things, and this all points we have to cover in the temperature as an ecological factor. And food chains, mainly in ecosystem, food chain is very, very important. Actually, in the food chains, 3 food chains mainly we have: grazing food chain, parasitic food chain, and detritus food chain we are saying. Grazing food chain is always starting with the solar energy, that is sunlight. Always grazing food chain will be consisting of producers, consumers, all these things we are getting. Producers, primary producers, and consumer, first tropic level, second tropic level, third tropic level, fourth tropic level we are saying. If you take the common example for the grazing food chain, plants are the producers. On the plants, insects are feeding. Then on the insects, frogs, and on the frogs particularly we can take a snake. On the snake, eagle. That means this example we can take first tropic level, second tropic level, third tropic level, primary consumer, secondary consumer, tertiary consumer, all these things we have to explain. And parasitic food chain always indicates actually parasites which we had, which is depending on other organisms. We can explain about the parasitic food chain. Common example we can take. Detritus food chain will be starting here. The producers are the dead and decaying matters particularly. And on the dead and decaying matters which are feeding earthworm. On the earthworm which are feeding frog, they snake, and then hawk. That example we can take. Clearly we can write about the food chain. Sometimes if they are asking, they may ask about the air pollution, water pollution, or they are the common topics. We can explain about this thing. And lastly, the zoology second year, as we are seeing about the 8 marks answers, compulsory from the 4 life cycles: entamoeba, plasmodium, eukaryoria, and ascaris, 1 life cycle we are getting. And from the ferritima, that is earthworm digestive system, earthworm reproductive system, compulsory 1 question we are getting. Epithelial tissues is very important answer. And last unit, that is the temperature ecological factor, food chain, compulsory 1 question. Very, very easy. If you are preparing the 2 out of this 4 concepts, 2 concepts if you are preparing, you can clear the long answer. Out of 4 you have to write 2.